We'll just wait for maybe a couple of more minutes and then we'll start. Okay, welcome to everybody. I think we can go live now if we aren't already. Are we already live? Whoever's there from NPTEL or Navisa, can you confirm?
Yeah, I'll just check in with them.
Yeah, thanks, Navisa.
I got a notification when I joined that the session is being live streamed. So I think.
Okay. All right. Great. So welcome all bright and enthusiastic learners out there. We are going to be discussing the week 6 lecture on basics of research design. Now, I know it's a bit of a heavy lecture, so it's not surprising there are not as many of you here as they normally would be. And it's not specifically about birds and ornithology. It's a more general introduction to research design in any kind of science, and maybe a little more particularly any kind of ecological science, by which I include behavior and evolution and so on. So we'll dive straight into it. I'll first look at the questions that came in on the Google form. There are only a couple of questions that are relevant over here. There's one question that asks, can you give a brief history of avian research in India with a timeline? That was a subject of the first week, and I think in the additional readings for the first week that you'll see on your dashboard, there are links to exactly what you have asked for over there. So please look at the additional readings for the first week. There's a question from Akshay. I'm going to read it out verbatim. does injecting a neutral change an individual's behavior? Is there a chance that certain individuals are responsive to it? So I think, Akshay, you're referring to the discussion on the controls in an experiment. And sometimes if you are, for example, with the people, if you're injecting you're doing a study on the efficacy of some injection, let's say an injection against pain, painkiller injection. And if you're studying the efficacy of that, then your treatment, of course, will be that pain, that injection of the drug. And then the question is, what is the appropriate comparison? And obviously, not injecting anything may not be an appropriate comparison, because just the very act of injecting the needle going into your skin may change your physiology and does change your physiology. And so then we do need an injection as a comparison. So an injection of something neutral, like saline, for example, saline that you get at the hospital when you're dehydrated. And the question is here, is there a chance that certain individuals are responsive to, for example, injection of saline? And it's quite possible that they are responsive to it. So we have to remember that we are comparing an injection of the active drug with an injection of saline or whatever else the control is. Similarly, if we're doing a playback experiment like described in the lecture, we're looking at how do birds respond to a playback of their own song, the song of their own species. And we are comparing that not to no playback or no sound, but to some other bird sound, because it's possible that birds respond to any sound or any bird sound. And so the key measure is the difference between the treatment and the control, in this case, the treatment being the playback of the song of their own species, and the control might be playback of a song of a different species. Or some people use as a control broadcasting just white noise at the same decibel level. So we always have to be aware in experiments that it's the comparison that matters, and we need to be very clear on what exact comparison are we making, what is the control, and why is that the control. So I hope that helps a little bit about why we're injecting this neutral substance. There's a question about what is local migration. So this is not really a question from me, I think. I think this is something that we had a guest lecture on, I believe, on migration.
Yeah, this
and
is Madhumita.
Madhumita, and I think that she would have given these examples. So she's not here, so let me see if I can say something about it. Local migration is just short distance migration versus long distance migration. That's the only thing. There's no very clear cutoff definition for this. For example, there's some within country migrants in India, the Indian pitta, Indian paradise flycatcher, that migrate between central and northern India on the one hand in the summer, and the winter they come down to southern India. So that's could be called local migration. We call it within country or within subcontinent migration. There's altitudinal migration, elevational migration, where the birds may move a very short distance, but in that short distance they move a very large span of elevation. So some of the high altitude birds in the winter, they come down the Himalayas and they come down to the lower altitudes. The same person asks, why do cuckoos migrate? Cuckoos migrate like any other bird, which is to escape harsh conditions and low food conditions, and to go to places which have better weather and food conditions. From where are they coming? This varies from cuckoo to cuckoo, which flyway they follow. So some of the cuckoos, for example, the common cuckoo that we have that breeds all across Eurasia and the Himalayas and has a small breeding population in central India, they winter largely in Africa, southern Africa, and so they don't follow any particular flyway. In fact, those flyways that Madhumita would have shown you some global diagrams of different flyways, and people usually put India within the Central Asian flyway. That looks as though all the birds, all the species that come from that particular part of Central Asia, they all winter in India, and that's not. I mean, that's just a broad generalization. It's not true for every species. And my suspicion is that those flyway diagrams, those maps, were made largely with shorebirds in mind, with the waders like sandpipers and greenshanks and godwits and things like that. In many cases, for land birds, those maps don't apply. So, for example, the red-breasted flycatcher, which all the red-breasted flycatchers in the world, which breed all the way across from Western Europe, so England, all the way to around Central Asia, they all winter in the Indian subcontinent. So the red-breasted flycatchers that breed in England are coming to India for wintering, and that you don't see in the flyway map. Similarly, we don't see those passage migrants that pass through India from Eurasia on the way to Africa. Those aren't depicted on the flyway map. So the flyway map is just it's a general, gives a general notion, but we shouldn't take it too literally for all species. And I'm just reading the last question in the Google form, and then I'll come. I can't see if there are questions there. In the meantime, please do type your questions in the chat box in Zoom. So there's a question about, actually, it's a question about one of the questions in assignment 5, which is about spectrograms and bird vocalizations. But this is not something that I can answer. It says there's no option to choose multiple answers. Anyway, I will look into this. I think the hint here is that there may be a mistake in the question or a mistake in how the question was depicted on the platform, but we'll take a look at that and get back to you. Okay, so I'm done with the questions in the Google form. Now I'll look at the chat box over here. Hello to all of you who've said hello. Very nice of you. During clinical trials, it's common that the sample size may reduce as the study progresses. For example, people may die, I'm imagining, or volunteers may decide to withdraw. So actually, if we are looking at, for example, the efficacy of a drug to reduce, let's say, cancer rates, and those typically those studies occur over a long period of time, and we are comparing that drug, the treatment with the control. So some set of people who've got some kind of placebo, some non-drug, or more often these days we have a new drug compared with the older drug, because we don't want to. It's a bit. You could imagine it can be considered unethical to have a control where cancer patients are given a substance that medically is known to have no medical effect, right? So that would be a classical control. So instead, typically we might give a newly developed drug to see whether it's more or less efficacious than an existing drug. And so as the study progresses over time, over years, you'll get some people whose cancer grows and doesn't subside, and they may die. And in fact, removal from, quote unquote, removal from the trial through death can be considered an outcome of the trial. That is, one of the things you want to measure is after 5 years, what fraction of people in the treatment are still alive, and what fraction of treatment in the control are still alive. So in some cases, it might be built into the study design. When volunteers decide to withdraw at a later point, that's actually a very bad thing. If it's under the control of people to withdraw, then we never know. People may tend to withdraw at higher rates in one or the other of the two, in the treatment or the control. And that can contaminate the results that you get. So for example, if the new drug has got more side effects, and because of side effects people withdraw, and those withdrawals are treated as though they are dead, for example, then you get the wrong idea. So I think typically, I'm not an expert on clinical trials, but typically I think you get all the details and so on of the people who volunteer for the different arms of the experiment, and you'd follow up with them and try and ensure that there is as little drop-off as possible. You definitely want to minimize the amount of drop-off. Not because of the sample size reducing. That can be a concern, but typically you need to have a large sample size, so a few people dropping out is not an issue, but more to guard against the possibility that people differentially drop out in different arms of the experiment. That would be the larger concern, I think. I hope that made sense, Gaurav. You can always ask a follow-up. Gajanan asked, if I want to study nest site selection by breeders, what criteria should I include in my research set? Okay, so I mean, all the criteria that we talked about, you've seen the theory, which I've tried to give you in the lecture. But of course, translating the theory into practice is not a trivial sort of thing. So I'll talk about nest site selection, not by waders, but by any bird, right? And so I want to know, basically, I want to ask what features of the habitat are birds choosing when they decide where to build a nest. And so the typical way in which people do this is they say, well, if birds were not choosing any aspect of the habitat, if they were just nesting at random, then what would an average nest look like? Not what the nest would look like, but what would the microhabitat or the habitat around the nest look like? So I can imagine, you can imagine throwing down random points on the habitat, going to those points and making some measurements of, let's say, tree height or leaf litter or amount of shade or horizontal cover, concealment, things like that. And then you go to the actual nests that you find and you measure those same things on those actual nests. For those actual nests, like concealment and shade and leaf litter and whatever else, distance to human habitation if you're worried about human disturbance. And then you typically compare what a random bird might look like, a bird that didn't have any selection, really, it just put its nest anywhere, compared to what the actual nest looked like. So this is, by the way, it's not an experiment, of course. This is an observational study. We're not manipulating anything here. We're just looking at how different are the actual nests, location of the nests from random locations, and in what way do those locations differ, and can we make sense of those locations? And this would apply, as I say, for any study of nest site selection. Archana asks, how does one identify transects and ensure that they are represented over the larger population? So this is, I think, Archana, we sort of talked about generally how one tries to attain representativeness. If you have a large sample size that you can use, large number of transects that you can walk, then one way is to just generate a random number, or rather generate, if you're putting these transects in a space, in a two-dimensional space, like on a forest or grassland, then you can generate a random X and Y coordinate from the range of X and Y coordinates that you have available to you. Put that on the ground, go to that place on the ground and start walking the transect. You also presumably would want a random direction to walk in, which you can choose between 0 and 360 or 359 degrees, assuming that your transect length is fixed. And as usual, if you can't walk many transects, you can only walk, let's say, 5 transects, then you want to ensure that those 5 transects are distributed in some representative way across your landscape. Otherwise, if you just did it using a random number generator, quite easily your 5 transects could be clustered in one part of the overall area you want to generalize to. And if not all 5, then 4 of the 5 may be clustered. That means you're over-representing one corner of the area and you're underrepresenting everything else. So then you might want to do either stratified or systematic sampling. So that's generally how you would do it, like you would just do with plots or quadrats or grids. The principle would be the same.
Okay, thank you.
Thanks for asking. Nafisa, thanks. That's a good idea. Well, I'll just come back to, I think, Gaurav's question. Yeah, you're thinking about COVID trials. Yeah, I agree, Gaurav. So dropout rates or people dropping out are a concern. This is particularly a case with experiments and clinical trials that involve people, of course. Now, we are, of course, in ornithology, so we are focused on birds. And birds can also drop out. So, for example, if you're looking at the effect of, let's say, a hormone implant, and people have done this, they've implanted little, how to put them, tablets, not tablets. What are those pills capsules, little capsules of hormones under the skin that slowly release, for example, testosterone or estrogen, because you want to look at what's the effect that these hormones have on birds. Like, do they come into breeding condition earlier than a control? Now, you do 30 and 30, 30 with testosterone implants and 30 without, and you're happily following them, and what you want to see is whether the ones with testosterone implants come into breeding plumage or start singing earlier than the others do. But let's say you start to lose them. I can't find them anymore. And I don't know why I can't find them. They might have died or they might have moved away from the area. But the problem is, if they died or moved away for some reason that's related to the implants, then that's contaminating my trial. If they died for other reasons like this natural mortality, some falcon or whatever came and ate some of them, which has nothing to do with the experiment itself, then it doesn't really matter because there's no bias in how they have dropped out of the experiment. But if it has to do with the experiment, then I really have to worry. And of course, the experimenter doesn't necessarily know why they've disappeared. So if there is a high rate of dropout, then it's possible the experiment kind of hasn't worked and one can't make much of the results. So that is a problem certainly in field-based studies where things can go missing. Certainly free-moving animals can go missing. This is less of a case when I'm following trees or looking at what's happening to grass in plots and things like that. Yeah, Nafisa, thanks from the discussion forum. Can generalization be made a requirement of study design? Yes, it is a requirement, but the degree to which you want to generalize is up to you. I may only be interested in this particular forest, right? I may only be interested in one particular forest, and so I'm surveying that forest, but I want to generalize from my transects or plots to that forest. Let's say the forest is 5 square kilometers. It's a small forest. I still can't survey every single square inch of that 5 square kilometer forest. I still have to sample within that 5 square kilometer forest. And if my universe or the population to which I want to generalize is that 5 square kilometer forest, then I have to ensure that my sampling is representative of that 5 square kilometer forest. Now, let's say that forest is a dry deciduous forest in southern India. And my intent actually is to generalize to dry deciduous forests in southern India. But there are many dry deciduous forests in southern India. Right? So just studying that one particular forest is going to tell me a lot about that forest, but doesn't necessarily tell me about the larger set of all dry deciduous forests in southern India, because I don't know whether they are the same or different from this forest because I have not been able to go and check. If I do want to generalize to that larger entity, then I'd have to survey multiple dry deciduous forests in southern India. Not all of them, obviously, because that's not possible. That's where the generalization comes from, but a representative sample. Or let's say I want to generalize to dry deciduous forests globally. That also can be your ambition. But then you can only do so if you sample in a representative way across dry deciduous forests globally. So you can't do one thing like sample one forest, dry deciduous forest, and wishfully generalize to everything else. Right? That's why your intent has to come first. What is it that you want to do? To what population do you wish to generalize? That determines your sampling strategy. Now, if you decide, you say, well, I want to generalize to all deciduous forests all over the world, and then after you work out some of the details, you realize, boss, because of visas and time and money, I can't do this. Then your ambition shrinks a little bit, right? Until such point, you say, okay, this is feasible. Or you can say, I still want to do it globally, but I'll do it through collaborators and I'll write to lots of scientists all over the world, blah, blah, blah. You can still do it. But the point is that you have to start with that larger sampling frame, the larger population, and then sample within it. And if there are results, the second part of the question, if there are results which are specific to a geographic area that cannot be extrapolated, and that's going to be true for most places, then you cannot extrapolate them. That's fair enough. But you only know that they are unique to that geographic area if you sample multiple geographic areas. If you only sample one geographic area, you have no idea whether it's unique or whether it's actually quite typical, right? So you're stuck. You have to start by defining where you want to generalize to and then proceed from there. What about yellow wattle lapwing local migration? So I don't know what that question is, so I will skip it. By the way, lots of birds show local movements, which we might call, and migration, by the way, migration is usually used for a regular back and forth movement. That is quite predictable. But you also have birds like for example, Brahmini starling is one such, which basically is, we think at least, it's fairly nomadic. So nomadic birds actually just move around and it's not predictable where they'll be at any given time. So it's raining there at some point, then they'll move to where the rain is because their grass grows and grasshoppers are there. If it rains in another place, they'll move over there. And birds have this uncanny ability to be able to sense some of these things. For example, I think it was cattle egrets. In Europe, they did a very nice experiment to show that cattle egrets can move large distances because they can smell where a tractor has either mowed the grass or has upturned the earth, has plowed the earth. And they can smell this over a distance of many kilometers, 10 or 20 kilometers. Because there's something in the air, the volatiles are released in the air that tell them. And of course, cattle egrets follow tractors and they like upturned earth and they like freshly mowed grass. And so they do that. So at that small scale, cattle egrets are following the resources as they are. It's not a predictable movement. So there are a few birds at least that show this unpredictable, more nomadic movement. In week 6, your part looks harder. Sure, harder than the other lectures? Yeah, I guess so. And if you think this part was hard, then steel yourself for the last couple of lectures of mine, which are about basics of data analysis. So those do get to be considerably harder, especially if you don't like maths and statistics. But maths and statistics are a necessary evil. I shouldn't say the evil, but they're a necessary thing for you to learn if you want to be in research. How does one distinguish between uncertainty and standard deviation? I don't know how. I'm not sure what the question is. Uncertainty and standard deviation are two different things. So standard deviation is measuring the variability in the data. Uncertainty, so just the variability in the data. I collect the body weights of 100 crows, and if there's a large difference between the lightest and the heaviest crow, therefore, then it'll have, that means there's lots of variation from individual to individual, and that will result in a high standard deviation. Uncertainty is how well or not I have estimated the mean, for example, the mean body weight of crows. So if my mean estimate, I think, is somewhere close to the population mean. Remember, I've only measured 100 crows, but the population of crows is, let's say, a few lakhs. So I measured 100 crows out of those. Now, how do I know whether that mean of that 100 is close to the true mean of all those lakhs of crows? That is what uncertainty is. And uncertainty is measured, as I've mentioned in the lecture, through something called standard error. Actually, I don't know whether I say standard error. It'll come in the data analysis lecture. I can't remember now. I'm getting confused whether I talk about here. But one of the graphs I show in this lecture is that if you measure the crows in an unbiased manner, that is, A, your measuring instrument is not biased, and B, if your 100 crows are a representative sample of all crows, then as you measure more and more crows, the mean of those, let's say 10 crows and 20 crows and 30 crows and 100 crows, the mean of those will get closer and closer to the population mean. So a large sample size always helps in reducing the uncertainty. Remember, this is the uncertainty in generalizing from the mean you got to the population mean. However, remember that if your measuring instrument is biased, that is, you have got a weighing scale that always measures 5 grams less, that's a bias. Or when I'm catching crows to measure them, let's say all the small crows are really smart and they don't get caught, but the large crows are slow and they're a little less agile and they get caught more, then my sampling is biased. 100 crows that I measure will tend to overrepresent the large fat crows that can't move very fast, and it'll underrepresent the small agile crows. I'm just inventing a reason, but that is a bias. And the big problem with that is, and I'm deviating a little bit from the question, the big problem with that is that you have no way of knowing that your sample is biased because you don't have access to the overall population of crows. You only have the 100 crows that you measure. So you have no way of knowing that whether the 100 crows are really representative or they are overrepresenting the large crows that can't move as fast. And so you have to think over and over and over again, what are the sources of bias in my sampling? Is it possible that my way of catching crows is biasing my sampling in some way or the other? and try and find ways to overcome that in this. Yeah, so that's your task as a researcher. Yeah.
Okay, I understand because I think I understood it wrong because I understood uncertainty as how far you move away from precision, but precision looked like mean, and then therefore uncertainty to me looked like standard deviation.
No, so
Now I understand.
uncertainty and precision are just the inverse of each other. You know, it's like saying how accurate am I versus how inaccurate am I? They're just the opposites. Uncertainty and precision are the opposites in this research design terminology. Okay, so one is the inverse. If I take precision and I do one divided by precision, that's uncertainty. One divided by uncertainty is precision. They measure the same thing, except they have the opposite kind of meaning, if you know what I mean. Yeah. So they're measuring the same thing. So variation is different. Variation in the sample is entirely different versus the precision or uncertainty of the estimate in relation to the true population parameter, in this case, the population mean, which I don't know. I'm trying to infer the population parameter from the sample. And how well can I infer is precision. How badly can I infer is uncertainty. You see, it's just the same thing, but just the opposite.
Yes, I understand. Thank you. Yes, your explanation has helped. Thank you.
Okay, great. Thanks. Gaurav says sometimes. Yes. Yes, in fact, Gaurav, that is true. If your sampling method or many aspects of your methodology, when you first think about them completely theoretically, before you go into the field or before you start collecting any measurements, you will have one conception. Oh, this is how I can go and do things. And then you go out into the field and start collecting data and you realize, I was mistaken. I can't do it this way. I can't select transects this way, or I can't catch birds this way. Or when I do my playback experiment like this, I run into all kinds of other problems, so I have to modify things. So you do have to modify it, right? You can't just stick with what you decided completely theoretically. But you can't mix the results from the pilot and the main part of the study. So it's really important, I think, in many cases, especially if you're doing or in your group that you're working with, if this kind of study is being done for the first time, there's no experience with it, then you have to budget in a pilot period. And that pilot is simply to learn how best to do the study. You would not and you could not combine the data collected in the pilot study with the data collected under a changed method. So I think you're absolutely right. If you take an entire year, as you are suggesting, to do the pilot, then yes, your first year data cannot be combined with the rest. But usually what you would try and do is you would do a pilot study quickly. You would do it, let's say if you have a 6-month master's dissertation to do, you do your pilot data collection in a week or in a few days to very quickly understand what the pros and cons are, what the feasibility is, and quickly adjust your protocol, your methodology. But once you've adjusted it and changed it, then you want to stick with it, because, as I say, you can't combine data collected under different methodologies. That won't be appropriate. So 3 cheers for pilot studies, but do them quickly, get them out the way until you can, and then settle down into the final protocol. Highly recommended, yes. Dr. Surjit asks, standard protocols, lab research work in ornithology. How do we establish a research center in our government college? So I'm not a lab biologist, so I'm a little bit lost about how to answer this question. Lab research also is very variable. So, for example, you might, in some lab research, you actually work with birds in the lab, right? So you'll have caged birds and so on. There are people, for example, in IISER Pune, who work on the neurobiology of song, of vocalizations in zebra finches. So they have a colony of zebra finches in the lab, and they do, and so they have to maintain that colony. I think they might breed them. Typically, if you have live birds in the lab, you try to breed them in the lab rather than keep getting fresh ones from outside. And then you might do various things with them. But there is other kinds of lab work. So, for example, you might go out and collect blood samples or fecal samples from wild birds, and then come back in the lab to analyze those samples. So, from fecal samples, you could get some idea about diet. You might get some idea about parasites. From blood samples, you can get some idea about blood infections. You might get an idea about people do some DNA sequencing and try and understand how different populations of birds in different parts of their range are related to each other, something called phylogeography. So there you don't actually have the birds in the lab. You have only samples from those birds. And then you could also take photos or take sound recordings of birds in the wild, and you could come back to the lab and analyze those photos or sound recordings. And so there's an entire spectrum of work you can do away from the field. As you can imagine, it's more complicated, both in terms of setting up as well as all the permits required to have live birds. It's a little less complicated to work with tissue samples or fecal samples. And it's presumably, I think, the least complicated to work with photos and audio. So depending on what it is that you want to do, there may be different requirements to set up a lab and lab work in your college. For the photos and audio, you just need a desk and a computer. So presumably setting up a research center of that nature in any place is the simplest thing. Again, both financially as well as from the permit point of view. So I think the first thing to ask yourself is, what kind of work are we interested in? What kind of, and why? Why is this important? Why is it important to now? What are the interesting questions to be asked? And the interesting and importance of the questions has to kind of interact with the feasibility of what that would entail, what answering the questions would entail. You might say, yeah, for this I need to set up a DNA sequencing facility, but is it feasible to set up a DNA sequencing facility? And if you decide not, then you may leave that question aside, even an interesting question. Or you can find ways around it. For example, you could say, well, actually, nowadays most DNA sequencing is done by commercial outfits. You just send the purified DNA or extracted DNA to a company, and they do and you tell them what you want, what kind of protocol, what kind of procedure, and then they do it. I'm not using the right terms because I don't do this kind of work. So there are many ways in which you can try and overcome also some logistical difficulties, either paying for somebody to do it or collaborating with somebody and so on. So again, you just have to start, I think, with what it is you're interested in and why, and then go from there to see what's needed. I don't know if that was a satisfactory answer. Okay. Yes, please do go ahead. Yeah. Yeah, Dr. Surjit.
Yes, sir. I am quite satisfied. Thank you, sir.
Okay, great. Thanks. Please do keep typing questions if you have. I'm just going to look if I've maybe missed anything. Okay, I don't think I missed anything. Nafisa, was there anything else on the discussion forum, or was just that one?
No, there's no other questions in the forum.
There's just that one question. Okay.
There's another question.
I saw that just now. Yeah, thanks. Yeah, Gaurav, this is a really nice, nice question. Yeah, let me try and think, come up with a specific example of this. Okay. Now, suppose I'm a researcher, okay, and I study sex differences in behavior. How males and females differ in their behavior, and I'm particularly interested in breeding behavior and the sort of things surrounding that. And I study one of these species which has so-called sex role inversion, like the greater painted snipe, where it's the female who defends a large territory within which there may be multiple males, and she mates with the one male, and then a male builds the nest and she lays eggs in that male's nest, and then the male goes ahead and incubates the eggs, and then she mates with another male in her territory and lays a set of eggs for him, and another male and lays a set of eggs for him, and so on. So a single female has multiple husbands, so to speak, within a territory. And of course, females, you might expect, are quite aggressive towards each other, because females try and steal males from each other because the more males a female has, the more eggs she can lay, and therefore the more offspring she can have. So let's say I'm interested in understanding how females allocate their time in defense of their territory. And you would imagine that if there's a strange female that comes in to establish female territory, that she would attack that female. But I think I have this idea in my mind that females gauge their response, they adjust their response according to how much they have to lose. So if I have a female, I've got only one male in my territory, then let's say at most the intruding female can take one male away from me. But if I have 5 females in my territory, an intruding female could, if she defeats me, can take away all 5 of the males that I have. So I have more to lose the more I have. Okay, I'm just coming up with this on the spur of the moment, so this may or may not make sense. So the experiment that I do, having had this idea, is I simulate a territorial intrusion. I either have a captive female in a cage, or I play back the sound of a female, unfamiliar female, and then I look at the response of the territorial female. Okay. Now, my prediction from the hypothesis is that the territorial female should respond much more strongly if she has more males in her territory, and a female who has only one male in her territory will respond much more weakly. I hope that makes sense to you. And I'm trying to gauge this through this experiment, either it's a playback experiment or putting a dummy or even a model female that is not a carving of an intruding female. Now, because I came up with the study, and because it's a favorite idea of mine that females adjust their aggression response depending on how much they have to lose, I will unconsciously be biased into I want the outcome to be the way that I thought it will be. Unconsciously, I want it to be, because then that'll make me feel fulfilled. That females with 5 males will respond more aggressively than females with 1 male. And so when I'm watching these females, in the course of the experiment, I will tend to again unconsciously score the behavior of females with 5 males to be more aggressive, for the same action. Right? The female moves ahead like that, and I have to score it on a rate of 1 to 3. On a scale of 1 to 3, I might score it 3. Whereas a female with only 1 male, even if she makes the same movement, I might unconsciously make a different decision and score it to be 1 rather than 3, because that is in accordance with my pre-existing understanding and bias. Now, one of the purposes of science or research design is to try and reduce those sources of bias as much as possible. So one way to do it is where the person who's actually looking at the behavior doesn't know anything about the study, right? Has not heard my hypothesis, does not know about my prediction, and is just scoring the behavior in the field blind to what I have proposed. Now, that's very hard to actually organize in a real sense. The students or assistants or whoever work with me, I would have told them some story about why I'm doing this work and so on. So they'll be contaminated by the same pre-existing expectations that I am. So, sorry, that was a very long example. So one way to overcome this is, rather than scoring the behavior in the field itself, you videotape all these responses to the intrusion experiment. And then I have multiple videotape files, and I don't label them like we normally would. You might say, 5 male female response on this date, and 1 male female response on this date. Instead, you name the files in a way that just a serial number or something like that. You have some Excel file somewhere else where you know which file refers to what condition. And now you can watch those videos blind to whether this was a 1 male female or a 5 male female, right? And even better than you doing it, because you might remember something about the habitat that was different, and that still may contaminate your the way in which you do it. Even better, now I can tell some completely different person. I can say, okay, here are the rules for scoring the behavior as more or less aggressive. Here is a set of females. Don't worry about whether those females are 1 male females or 5 male females. That's not your concern. Just watch the video and score the behavior according to these rules that that we've come up with. So that person is then truly blind to which female is which. And then if I find the results in the direction as expected, I can be particularly confident that indeed 5 male females actually are more aggressive, and that result is not contaminated by my pre-existing bias, and therefore any change or bias in the way that I score the behavior. And this kind of thing generalizes to any kind of research in the field, where when you're in the field, you know what condition you're in. Like, for example, if I'm I'm studying coral reefs, and I have studying two kinds of coral reefs, one where there's a lot of fishing and one where there's no fishing. And I have an idea that fishing reduces herbivorous fish, which increases the amount of algae that grows on the corals, which stops the new corals from, baby corals from settling and growing, right? And so I'm in the water there, and I'm looking at a quadrat, and I'm scoring, okay, what area in this 1 meter quadrat is covered by coral, and what area is covered by algae. And even a simple measurement like that, my biases can shift the balance a little bit. But instead, if I take a photo of every quadrat, 1 meter quadrat, in the high fishing place area and in the low fishing area, take multiple photos like this. I come back home to the lab, I scramble those photos. I don't know anymore, or my assistant or my student doesn't know anymore which photo is which. The decoding of the photos is only in an Excel file somewhere, which nobody's looking at. Then I can be sure that whatever difference I find is not because of bias in favor of finding a difference in one way or the other. Okay, so one very long example, one short example, but just to indicate to you that we have to fight against, and it takes work to fight against the pre-existing biases we might have. All of us have these biases. We all have some expectation, and those expectations of the results can very easily contaminate the results as well. I hope that made sense. Oof. Okay, Gaurav, another Gaurav. This is not really, well, maybe it is in relevant to us. How can you observe breeding without disturbing it? Typically, the best way to observe breeding without disturbing is remotely through either a camera trap, which takes photos when there's movement, right? So whenever there's movement, then it fires. That's to save space and battery power, or through just video, where and then you can watch on a screen either in real time or later. That's the best way without disturbing. Of course, in the past, before these technologies, people have used other methods. You use hides. So a hide is like a little tent that you install at some distance from the nest with a slit through which you can observe, and the birds easily get used to the tent, and don't seem to realize that a human enters the tent, but the human doesn't go away. Therefore, the human is still inside. Most birds don't seem to realize that, and so once they, in a minute or two, they see there's no movement, there's no person, and they get used to it. So a hide is another good way of observing breeding. Ooh, what is this photo, Sushila? Oh, this is a house sparrow nest on a fan. Oh boy. I always worry about electrocution when that happens. 4th time. Yeah, that's really interesting. And of course, if something is nesting in your house itself, there's lots of interesting things to observe. Like, for example, what is the role of male versus female, and do they both incubate, and they both feed the young, and to what degree? How long does it take from incubation, from egg-laying to hatching? How long does it take from hatching to the chicks leaving? What kind of food are they bringing? All that kind of thing. Lots of cool stuff on it too. Okay, we are nearing the end of our session anyway, and I think the questions have dried up. I think my painted snipe example has exhausted all of you. So if there's no further questions, we can wrap up. Any final thing, Nafisa or Devika?
No, that's all.
Yeah. Okay, then I'll thank you all and take you leave. I hope you're enjoying the course. I know some of it is tough going, but I'm hoping it's worth it. And I think we're halfway through the course, so another halfway to go. You made it 50%, you can make it all the way till the end. So with those words of encouragement, I leave you there. Nice to see you over there. The advanced course, at the moment, there's no plan for an advanced course. We are trying to see whether we can do an advanced course, of course, would be multiple ones, no? Because there could be an advanced course in bird acoustics, it could be an advanced course in whatever behavior, and an advanced course in migration. You can't have a single advanced course because you have to decide which direction you want to pursue. So that takes a lot of work to set that up, where every year we resolve to do it, but then another year passes and we haven't. So don't hold your breath for an advanced course. There are other resources across the internet from other parts of the world. Please don't hesitate to use those as well. But we will try and organize 1 or 2 advanced courses here as well. Yeah. Thanks a lot and have a good weekend. See you next time. Yeah. Bye.
Thank you.